Hello, dear students. You are welcome to my channel about microbiology tutorials by Anshul Kumar. Today, I am going to discuss about the topic regarding pathogenesis and molecular determinants of virulence in mycobacterium tuberculosis. As we all know, that mycobacterium tuberculosis is one of the most severe diseases which remains in the host cells for a longer duration of time, and most of the chronic illnesses is caused by the mycobacterium tuberculosis, and the major the organ which is affected are the our lungs. And one more factor about mycobacterium tuberculosis is that its spread or its virulent properties or pathogenesis depends upon the immune immunogenicity of the individual, or can say immunocompetence of an individual. And if the person's immune system is strong enough, the infection subsequently subsides, or if the immune system lowers down due to several causative factors, the disease may flare up, and the only target organ cannot it's not only the lungs, but it disseminates to other body parts, included the meninges, bone, and body fluids, genital tract. So, especially in the developing countries, it is one of the major factors which causes morbidity, increasing morbidity and mortality, and one of the major factors responsible for this increase in the prevalence rate existing in the developing countries or poor countries is that lack of awareness among individuals, nutrition deficient diet, or we can say malnourishment, and maintaining a healthy lifestyle. And it is a bacterial infection, and it is one of the some of the important few bacteria, I can say, that triggers the cell-mediated immune response in our body. Means that the cells involved, immune cells, mostly the T lymphocytes and not B lymphocytes. So today I will discuss in detail about the mode of the mechanism of pathogenesis and what are some of the determinants based at its molecular level. Here, mentioning the molecular determinants is important because when we say molecular determinants, it means that such pathways or such factors which have been taken out on the basis of its genetic makeup of a mycobacterium tuberculosis. So, as the gene of mycobacterium tuberculosis is red, and the different parts of the gene which is responsible for synthesis of certain proteins or enzymes, which causes its increase pathogenesis or enhances its virulence properties, is detected, and it becomes quite possible to an extent to treat these infections. So let's start today's lecture. So Mycobacterium tuberculosis is the causative organism for the disease called tuberculosis, and it's one of the biggest killers among the infectious diseases. Despite the worldwide use of live attenuated vaccine, BCG, and anti-tubercular drugs, in this, on the basis of anti-tubercular drugs, though different regimen has been advised or have been developed for treatment of Mycobacterium tuberculosis, but since this the regimen is a very long period, depending upon the severity of the infection, it may the medicine or the antimicrobial drugs can be have to be taken for a long period of time, even for a year. So usually, if the regimen is not properly supervised, patients in midway leave the drug which has been an anti-tubercular drug as soon as they feel better. And it is one of the greatest mistake from the patient, because though they have been they feel that they have been recovering, but not the all the bacteria has been eliminated from the host cells. And since they are on the treatment, midway leaving them develops helps in the mycobacterium bacteria to develop resistance against those drugs. And then it becomes quite a great menace, because as soon these bacteria, multi-drug resistant bacteria, start to multiply, and patient again become ill or start showing symptoms of tuberculosis, then the previous drug which has been advised or has been given to the patients become resistant, and then it becomes a upheaval task or a very difficult task to treat such type of MDR-TB. So it is also, it should be kept in mind that when a patient is undergoing treatment for any type of tubercular infection, it is necessary to properly supervised under the aegis of an experienced person. And that's why in our country, DOT system has been introduced. That is the directly observed therapy under the supervision, to check the patients which are undergoing treatment that they are taking their medicines regularly. So coming to our topic, that what are some of the important general characteristics of the bacteria, Mycobacterium tuberculosis. So they are aerobic bacteria, they are non-motile, non-capsulated, non-sporing, acid-fast bacilli. If you would be knowing, or if any the medical graduates would be knowing, that acid-fast bacilli means that they resist decolorization during the staining procedures, even with strong acids. So they are called as, they show acid fast due to the presence of mycolic acid, a waxy substance in their cell. Then they are slow growing, that is their reproductive period is higher. They multiply usually or reproduce in 20 hours in comparison to normal bacteria who take 20 minutes to multiply. So if they are cultured, then it may take even from a week to a month to for their growth in the artificial medium. They are obligate parasite, that is, they are strictly depend upon their survival on the yeah, from the nutrition taken from the host, and they are opportunistic pathogens. Some of the important antigens present on the mycobacterium tuberculosis is mycolic acid and Vaxd. Now coming to the pathogenesis of tuberculosis. So some of the important source of infections are patients suffering from open TB, then live cultures and clinical specimens. So we can say that it is one of the highly contagious disease which can spread from one patient or from one person who may be a carrier to another patients, or if a person comes in direct contact with the bacteria. And the mode of transmission is the through inhalation of aerosol droplets. So aerosols are the tiny droplets which persist in the air for a longer period of time. And if a patient comes in contact with these aerosols, and if it is inhaled, so if and the bacteria gets a suitable host, they may easily colonize them there. They can easily colonize these bacteria in the host cells. What are the early events in there? So during coughing, sneezing, and talking, a varying number of droplets are expelled from the body. These droplets become aerosols, which are 1 to 5 microns in diameter and are inhaled. These droplets first come in contact with the these droplets come in contact with the these droplets first come in con these aerosols comes in contact with resident macrophages, alveolar epithelial type 2 pneumocytes. That pneumocytes are those alveolar epithelial cells which can infect and grow in pneumocytes, and dendritic cells, all antigen presenting cells and activated T cells. Since dendritic cells are migratory, unlike macrophages, they may also play an important role in dissemination. Mycobacterium tuberculosis and macrophage interaction. The bacteria are phagocytosed in a process, which is the characteristic feature of a macrophage, and that is inherited by bacterial contact with macrophage nanos complement receptors. Then second comes the surfactant protein A glycoprotein found in alveolar surface, can enhance the binding and uptake of MTB by upregulating mannose receptor activity. Surfactant protein D, similarly located in alveoli, inhibits phagocytosis. So two other the immune mechanism in addition to macrophage activation include the surfactant protein A, which is a glycoprotein found on alveolar surface, and surfactant protein D, which is both of them are present on the surface of alveoli. Next important is the mechanism, defense mechanism are human toll-like receptor 2 also play a role in uptake, which is a type of receptors present on the cells. And in entry into host cell macrophages, MTB initially enters in an endocytic vacuole called phagosome. If the normal phagosomal maturation occurs, phagosome-lysosome fusion, these bacteria can encounter a hostile environment that includes acid pH, reactive oxygen intermediates, lysosomal enzymes, and toxic peptides. If proper phagolysosomal fusion takes place, so obviously the aim of the any phagocytes or macrophage is to engulf those pathogens And with the help of lysosomal enzymes, they break down these they break down these products or bacterial antigens into smaller products, and then they exocytose them. So usually here the characteristics of mycobacterium tuberculosis is to escape this mechanism of phagolysosomal activity, and usually they reside in a latent form, or we can say a dormant stage, in these phagosomes. So we will discuss it in later. Here what happens. So when the normal phagosomal maturation occurs, that is the phagosome lysosome fusion, these bacteria can encounter a hostile environment that includes an acid pH, reactive oxygen intermediates, lysosomal enzymes, and toxic peptides. As macrophage killing of bacteria occurs in the phagolysosome, this is avoided by the mycobacteria. And the procedure which is applied by in for their survival is that basically they prevent the increase in calcium ions. And this elevate and how what the increased level of calcium ions during this phagolysosomal activity do, that elevated calcium ions were associated with phagolysosome formation, and calcium and can also stimulate many host responses to infection, that is respiratory burst, nitrogen oxide, and cytokine production. So all these factors lead to the destruction of the bacteria. Thus, preventing increase in calcium ion levels would help Mycobacterium tuberculosis avoid this host defense mechanism. Selective advantage of Mycobacterium tuberculosis of staying in an early endosome is that there would be less host immunosurveillance by CD4 plus T cells. So there is decrease in the expression of MHC2 complex and presentation of bacterial antigens in macrophages after MTB infections. The mechanism by which virulent mycobacteria prevent phagosomal maturation is not known. But in normal maturation of the mycobacterial phagosome, there is a successive recruitment of RAB proteins. That is, RAB5 associates with early endosomes, and RAB7 is found in later endosomes. So if MTB phagosome that does not contain RAB5 does not recruit RAB7. Major events: infected macrophages in the lungs through their production of chemokines attract inactivated monocytes, lymphocytes, and neutrophils, none of which kill the mycobacteria very efficiently. The granulomatous focal lesion composed of macrophages, derived giant cells, and lymphocytes begin to form. The process is generally an effective means of containing the spread of bacteria. As cellular immunity develops, macrophages located with bacilli are killed, and this results in formation of caseous center of the granuloma, surrounded by a cellular zone of fibroblasts, lymphocytes, and monocytes. Although MTB bacilli are unable to multiply within the caseous tissues due to acidic pH and low availability of oxygen, so the bacilli may remain dormant but alive for decades. So this is the most important step, that even after being phagocytosed by the macrophages, they escape that immunosurveillance procedure, and they are left unrecognized by the immune cells. And even these cells are recognized through the production of chemokines, attracted inactivated monocytes, lymphocytes, and neutrophils are not capable enough to kill this mycobacterium, which gives it an added advantage for their survival within the alveolar cells, or we can say inside the lungs. And as the cellular immunity slowly develops, though macrophages loaded with bacilli are killed, and this results in formation of caseous center of the granuloma surrounded by a cellular zone of fibroblasts, lymphocytes and monocytes. Although in such cases, MTB are unable to multiply within the case's tissues due to acidic pH and low availability of oxygen. So the bacilli may remain dormant, but it can get activated after as soon as the immune system of the body fades or subsides. It is similar to the formation like as the spore forming bacteria which forms a spore form outside the body, or we can say under unfavorable conditions, and as they get a suitable host, they develop into a vegetative form. So basically the difference is that the spore forming bacteria, they are in sporulated form outside the cells. But here, though it is a non-sporing bacteria, but it is remains in dormant. Dormant means that it is not multiplying. It remains in an inactive state till it waits for an opportunity that as the immune system of the body will uh, will decline, and it will enhance its activity by converting itself into an active form. So the strength of the host cellular immune response determines whether the infection is arrested or progresses to the next step. When the host immune system weakens, the granuloma center can become liquefied by an unknown process and then serves a rich medium in which mycobacteria can replicate in uncontrolled manner. At the point, viable MTB can escape from the granuloma and spread within the lungs, which is also called as active pulmonary tuberculosis, and even to other tissues via lymphatic system and blood. That is called as the miliary or extrapulmonary TB, causing secondary or post-primary tuberculosis. Now, what are some of the important factors which helps or contributes to the success of this pathogen? So till now we have already discussed that how cleverly or how efficiently it have the capacity to survive even the host immune defenses are strong enough to contain it. And there are several factors which acts to which are the helpful in helping for the survival of this bacteria even in the dormant form for the longer period of time. So its ability to survive the redox state, that is the oxygen stress conditions, or we can say deficient oxygen conditions, because it is an aerobic bacteria, manifested by the host and its capacity to synchronize its metabolic pathways and expression of virulence factors. Two component proteins, namely DOS-S and DOS-T, are employed by MTB to sense changes in oxygen, nitric oxide, and carbon monoxide levels, while YB3 and anti-sigma factor RSRA are used to monitor changes in intracellular redox state. Using these and other unidentified redox sensors, mycobacterium orchestrates its metabolic pathways to survive in nutrient-deficient, acidic, oxidative, nitrosative, and hypoxic environments inside granulomas or infectious lesions. So they make a change in their metabolic pathway as such that under such stressful conditions, they are able to or they are capable to survive within the granulomatous tissues or infectious lesions. And though it may be in a dormant state, it can be a dormant state, and it can wait for a longer period so that these as soon as the favorable condition will exist or come, and it will start multiplying. In addition to these, some of the important factors, mycobacterium employs a battery of protective enzymes such as superoxide dismutase, catalase, and alkyl hydroperoxidase and peroxyredoxins to detoxify. And here SOD, CatG, and EHPC are the gene or the allele genetic part which help in the synthesis of these enzymes. So if CatG, through CatG gene, they are able to synthesize catalase, AHPC, alkyl hydroperoxidase, and so on. To detoxify and neutralize these redox stresses, SOD is a metalloprotein produced by prokaryotes and eukaryotes to detoxify superoxide radicals. The enzyme dismutases oxygen into hydrogen peroxide and molecular oxygen and has been shown to contribute to the virulence in a number of pathogens, including MTB, which have two genes, that is the SODA and SODC. Tuberculosis virulence factors. The virulence of MTB can be measured during the infection of macrophage and animals using several different assays, and various strategies have been developed to make mutations in MTB genes. Cell secretion and envelope function. First is the culture filtrate proteins. Culture filtrate proteins associated with mycobacterium tuberculosis, primarily identified through research into vaccines, virulence factors, and diagnostic tools. These proteins are actively secreted by the bacteria, often through the ESX1 secretion pathway. Major culture filtrate protein of MTB are CFP10, encoded by the RB3874 gene. It forms a tight 1:1 complex with PS86 and essential for MTB virulence. Then comes the ESAT6 6 kilodalton early secretory antigenic target, encoded by the RB3875 gene. This is a major virulence factor that forms a heptodimer with CFP10 and is crucial target for TB diagnosis. MPT64, encoded in the RD2 region, used in rapid detection of MTB. MPT59, a highly expressed secretory antigen. MPT51, identified as a frequently recognized cellular response antigen. CFP21, identified as a differentially recognized antigen, particularly in non-progressors. RB3618 and RB3621C, evaluated for serological test alongside ESAT6. And then comes the P and PP family proteins. Multiple members, E8, PP34, are secreted and play roles in virulence. Bacterioferritin, isolated from culture filtrate. So these are the isolated proteins from the cell secretion pathway and from the envelope, which are responsible for the virulence. And major changes in these genes can help in developing the vaccine development. Usually these are used in the vaccine development as to determine some of the major virulence factors. So these are some of the important factors. Then comes the AHPC, is a member of the peroxyredoxin family that detoxifies organic peroxides into less reactive alcohol derivatives and confers protection against both oxidative and nitrosative stress. AHPC mutants show an essential role in the resistance to host oxidative agents in the early stages of infection. Deletion of genes that encode methylene sulfoxide reductase, MSRA, MTB proteasome, PCR, BA, nucleotide excision repair, UBRB, and F420 biosynthesis, FBIC, are also hypersusceptible to RNS. 5 PRX enzymes have been identified in MTB, that is EHPE, TPX, EHPC, BCP, and BCPB. Now coming to the next, that is the cell surface components. So here ERP, that is the exported repetitive protein, or RB, secreted by RB3810 gene, is approximate equivalent to 28 kilodalton surface associated protein found in the cell wall and capsule. It contains a carboxy terminal hydrophobic domain that anchors it to the surface. ERP is involved in intracellular replication and maintains cell envelope integrity. Then comes the mycoserosic acid synthase or MAS, a membrane-associated protein involved in the synthesis of thiocyrrole dimycosyrosates, that is the PDIMs in short, which are crucial lipid components of the outer membrane. Then FAD-D26 fatty acid synthase synthesized by RB293 gene 2930 gene, an enzyme essential for the synthesis of thiocyrrole dimycosyrosates PDIMs. PDIMs are critical virulence lipids located on the cell surface that assist in post-immune evasion. Mycobacterium membrane protein large, that is MMPL7, is a resistance modulation division family transporter required for the export of the virulence lipid thiocyrrole dimycosyrase in Mycobacterium tuberculosis. Then some of the important enzymes which are involved in cellular metabolism is the lipid and fatty acid metabolism. One is the ICL. So ICL is the important enzyme which is involved in general cellular metabolism, and it is a it is an important enzyme which is synthesized by RB0467 gene. Then comes the LIPF, synthesized by RB3487C. Then FAD-D33, synthesized by RB1345. Then comes phospholipases C, synthesized by RB2351C, RB2850C, RB2349C, RB1755C, then PLCA, PLCB, PLCC, and PLEDG. Then comes the PAN-C and PAN-D, which is synthesized by RB3602C, RB3601C, and PAN by gene. Next is the important is the amino acid and purine biosynthetic genes. So basically, the important amino acids and purine biosynthetic genes are LEUD, synthesized by RB2987C, then TRPD, synthesized by RB2192C, then ProC, RB0500C, RB0500, then PURC, which is synthesized by RB0780C, is, I beg your pardon, RB0780. Then comes the important gene which is responsible for the metal uptake. And here MGTC, RB1811, then MBTB, RB2383C, then comes the IDE, RB2711. So this is the metal uptake gene, metal uptake proteins in the present in the Mycobacterium tuberculosis and synthesized by the following genes which I have mentioned. Then comes the anaerobic respiration and oxidative stress proteins. So this is one of the most important proteins which helps in its survival during the oxidative stress conditions. And already we have mentioned in our previous slides. Again, I am mentioning it that there is one is nitrate reductase synthesized by RB1161, then CATG, RB1908C, then catalase peroxidase, enzyme synthesizes catalase peroxidase, then AHPC, RB2428, then SODA, RB3846, SODC, RB03842. Then comes the transcriptional regulators. Transcription regulators are some of the important, they are called as sigma factors, which regulate the transcription of these proteins and regulate whether the protein, whether they will remain in the dormant form, whether it will synthesize further, convert themselves into from the DNA to RNA. So these regulate the conversion of DNA or the genome to the RNA. So what are those factors? The sigma factor, sigma A, RB synthesized by RB2703, then sigma F, RB3286C, sigma E, RB1221, sigma H, RB3 223C. Then comes the response regulator, that is PHOP, synthesized by RV0757, PRRA, RV0903C, PRV. Then comes the MPRA, that is RV0981. Then other transcription regulators are HSPR, synthesized by RV0353, and YB3RV34 3416. So these are some of the important regulators, or we can say the proteins or enzymes synthesized by the genomic sequences present in the Mycobacterium tuberculosis, and they hold a very important role in importance in the diagnostic, from the diagnostic aspect, and also from from the vaccine development. So in the next lecture, we will discuss about all these some of the important role of these regulators or proteins synthesized, and how they influence the multiplication or growth of these mycobacterium. So with this, I conclude my today's lecture, and I hope that the students would have definitely benefited from my lecture. And if they have liked it, or if they understood it, please like and do subscribe my channel, so that I may keep bringing these videos for you in future also. Thank you.